Hello, I'm Jan Leiomyosarcoma.
I'm Gayatri Menon.
Welcome to MedNews Week Conference, where we feature presentations by medicine's global leaders. Today, we have an amazing keynote speaker in Dr. Seth Pollack. Dr. Pollack is the director of the Sarcoma program at the Laurie Cancer Center of Northwestern University.
In today's presentation, Dr. Pollack discusses the latest in sarcoma therapeutics.
Did you know that Dr. Pollack is an internationally recognized leader, physician, researcher, and speaker on sarcomas? He has served as the chair of the National Leiomyosarcoma Foundation Grant Review Committee and received numerous national awards, including the Sig Cole Legacy Award, the Northwest Sarcoma Foundation, the V Foundation Translational Research Award, and the Sarcoma Alliance for Research and Collaboration Career Development Award.
He's truly a dedicated advocate and a leader in the field of oncology and clinical trials.
So let's tune in. Let's tune in and learn from this great global leader.
So how can we overcome this problem of inhibitory macrophages in the tumor microenvironment? Well, one thing we were really interested in is whether we could use a toll-like receptor to alter the microenvironment. We know, so toll-like receptors are part of the innate immune system. And when I say innate immune system, again, for those of you, because I know there's so many people watching, if it's been a while since you did your immunology, we talk about the innate immune system in contrast to the adaptive immune system. The adaptive immune system are the cells like the B cells and T cells that have super specific receptors that are going to attack a given pathogen, like a T cell that's going to be specific for a virus, or a B cell that's going to be produce a specific antibody. Toll-like receptor is part of the innate immune system because it's there to activate the immune system, and it's fixed. And in whatever context that you have, the cells have toll-like receptors that the immune cells will have toll-like receptors that are potentially activating, and toll-like receptors are found in pathogens. So these are signals that the immune system has developed to develop specific pathogens. And we were interested, we decided to use a toll-like 4 receptor agonist, and this one's specific, called GLA, is from the bacterial cell wall. So it's a component of the bacterial cell wall that we injected into tumors, and we did it concurrently with radiation to see if we could reverse this macrophage change that we had observed in this prior study, to see if we could make the macrophages in those tumors become activating, because that's very well established that toll-like receptors can change inhibitory macrophages to activating macrophages. And this was a phase 1 trial. So because it was an experimental trial, we did this on patients who had metastatic disease but had an indication for radiation. So they had metastatic disease but an injectable tumor that was causing them pain and discomfort, and which the tumor really necessitated, or at least the patient wanted radiation to help alleviate their symptoms. And the tumors that were injected did really well. None of these tumors grew significantly. A majority of them had nice responses, at least some tumor shrinkage, or a response that was significant. As I said before, we had a wide variety of tumor types. So this was a small study, so we couldn't really pick out which sarcoma subtype was having the benefit. And in some of the patients, because these were patients with metastatic disease, we were able to follow multiple tumors. So some of the patients had tumors that were radiated and had the injection, had radiation, but were in places where it wasn't safe to inject, and had tumors that were not radiated or injected. And you could see the patients like that, the tumors treated with both radiation and the toll-like receptor did better, right? So you could see they had a deeper response compared with just the radiated tumor, and clearly the unradiated or injected tumors did worse. And this graph over here illustrates the tumor size kind of hammer that point home. And you could see these CT scans that really show this very clearly. One question that people have been asking is whether these tumors themselves have toll-like receptor 4, and they do sometimes, but that didn't seem to be related to the response. Of course, there's a small cohort, but this study was modeled a little bit after some of the studies in hematologic malignancies, especially in lymphoma. There's a great researcher and scientist, Ron Levy from Stanford, who's done a lot of work with TLR9 and lymphoma. And in that case, the lymphoma cells themselves have TLR9, but that's not what we're trying to do here. Here we're trying to activate the immune cells that are in the tumor. Of course, there were some tumors that had a lot of TLR4, but that didn't seem to correlate with response. So you can see here are some of the best responses that we saw. One of them had very little staining of the tumor cells. They had immune cells in their tumor that stained positive, but the tumor cells did not look like they were staining for TLR4, whereas some of the other tumors did have some staining in the tumor. In the patients who had nice local responses, they had a big increase in the T cells, and they had an increase in the clonality as well. And we'll talk a little bit more about that as well. Here's some nice some MIHC staining that is showing the increase in cellularity, but also the increase in CD4 and CD8 staining that we see following the TLR4 injections along with radiation. Now, let's talk about clonality. So this is the graph that I showed you about before. This is in one of our patients who had one of the best responses. They had a huge increase in the clonality of their T cells. You can see here. And what I find, so here we're illustrating it, and you can see some of the specific clones that developed on treatments. This is from TIL that my lab is very good at growing out TIL, at least for analysis from these tumors. And you can see one thing that's so interesting to me, and I'm really interested, maybe some of you guys have ideas out there, and let me know. One thing that's just so interesting is that these T cells that were some of the biggest expanded T cells, they weren't just one single clone. What we saw is that there were a variety of gene rearrangements that resulted in that clone. Likewise, they didn't, you have different V beta gene families that can be cut up and comprise a T cell receptor. We saw different V gene families that comprise these T cell receptors, which is just so wild to me. And I don't know that that it's been seen in these sort of expanded clones before. Of course, we know that these sorts of TCR clones exist, but in the context of a cancer treatment, I think this is really something that's unique. And I'm really interested in these sorts of T cell receptors because I think that in my opinion, there's something about the fact that you have so many gene rearrangements converging on the same T cell receptor that they must be important, right? They must be for something specific and some sort of antigen that that is such that the immune system is primed to hone in on it. When we did single cell sequencing, we used a single cell platform that allowed us to really focus in on some of these clones. We found that they had a Th1 phenotype. These clones that were very expanded. So these were CD4 cells, and they had a Th1 phenotype based on the single cell sequencing that we did. We also looked at the PBMC clonality in these, and we saw that there was an increase in the PBMC clonality in the patients who had nice responses, and that a lot of the overlap in the expanded in in the expanded clones in the PBMC had a lot of overlap with the pretreatment TIL. So I'm really excited about these results, and I think that there's going to be a lot of ways that we can apply it. And, maybe in the question and answer session later, we could talk about kind of how we can kind of exploit some of these findings. But TLR4 is not something that you can just inject IV. It's too dangerous. It's too inflammatory. So we're also really interested in how we can manipulate macrophage phenotype using systemic treatments. And one of the things that we've been interested in is trabectedin. And trabectedin is a very old drug. Now it's was developed sort of right in the wake of Taxol. So Taxol was discovered. Of course, Taxol is the chemotherapy that comes from the yew tree. And after Taxol was discovered, there was a sort of a gold rush of people looking at natural compounds to see if any of them had anti-tumor activity. And trabectedin was discovered in a sea urchin through a high throughput assay. So basically they looked at the sea urchin, they looked at all the different compounds in the sea urchin, and they tested all of them against cancer, and they came up with trabectedin as an effective agent. And they didn't really know how it works at first. And through additional studies, we found that it binds in the DNA's groove and can prevent cell division through that. But we found that it's been shown that trabectedin is also very toxic to macrophages. So it has probably a primary effect, which is directly on tumor cells, but it also binds and prevent to macrophage DNA and prevents and kills the macrophages that are in the tumor. And that's probably an important part of the effect. So this is a great Cell paper where they looked at tumors and they found that, so this is a tumor. If you look at figure B here, the untreated tumor grows a lot. If you treat with trabectedin, that kills a lot of the tumor and you get the red curve. But if you use trabectedin and you also infuse this mouse tumor with the macrophages, you reduce a lot of the efficacy of the trabectedin. So like half of the effect in this mouse model of the trabectedin is coming from killing the macrophages, right? So there's some effect still. The trabectedin is acting on the tumor, but some of the effect is from killing the macrophages. And we looked at, so we did sequencing of tumors that were in patients that were treated with trabectedin in our institution. And we found that if you had a gene signature with the patients who had this M2 gene signature did much better than the patients who had this other macrophage gene signature. And the same thing with myeloid-derived suppressor cells, suggesting that there's a really important link between the myeloid cells and their response to trabectedin in our patients. So we combined trabectedin with this PD-L1 inhibitor avelumab in a phase 1/2 trial in liposarcoma and leiomyosarcoma. And my philosophy is that it would probably be a good combination, maybe even a better combination in some other tumor types, but liposarcoma and leiomyosarcoma are the two tumor types in the US where trabectedin is FDA approved. So we wanted the on-label indication for trabectedin. And there was a dose escalation and then a phase 2, a wide variety of patients within these two tumor types. And patients could have had a variety of different lines of therapy, up to 5 lines of therapy. So some of these patients were quite refractory. And we had a number of patients with really, really durable responses. And I thought this was really encouraging. Now, the primary endpoint for the study was response rate, and we didn't quite meet the response rate endpoint, but which I think is unfortunate, because I would look at a patient like this patient that's going out beyond 2 years these couple of patients in blue who had stable disease, but these are certainly successes because these are aggressive cancers. They tend to have overall survival of less than 2 years in the metastatic setting. So if you have a patients that are doing this, that's a success. And we saw a 6-month progression-free survival of over 50%. So by some metrics, this was a really effective combination, even if it didn't meet the primary endpoint. And that's why I do hope there are future efforts to, and I think probably there are some additional things, and we could talk about this too, maybe to even make this combination even more effective. And when we did TCR sequencing of these patients, PBMC, so this is pre-treatment PBMC, the patients that responded better to trabectedin had who had partial responses had so this is pre and post treatment, had higher clonalities, both pre and post treatment. So if you had a partial response, you were unlikely to have a low clonality. So, you know, they didn't quite not overlap. So you could still have an intermediate partial clonality and have a partial response. But these patients who responded had higher clonalities, suggesting a T cell repertoire. Remember, this is in peripheral blood now, but that their peripheral blood T cell repertoire that's skewed towards a specificity against tumor antigens. There's also a large body of literature showing that doxorubicin can be an immunosensitizer. And I'm talking about it here because, you know, one of the reasons why we got interested in doxorubicin was because it kills macrophages. And we did a number of experiments looking at the tumor immune microenvironment in various models and showing that doxorubicin did a good job killing the macrophages without having a big impact on the T cells. But even before the age of immunotherapy, people were using doxorubicin to kill tumor and cause antigen presentation. So that's one reason why we think that doxorubicin can be a really great addition to immunotherapy. And of course, in sarcoma, doxorubicin is the standard frontline agent for metastatic soft tissue sarcoma. And that is despite the fact that it really isn't a good drug. So you can see here, it's the frontline agent. There's nobody who's questioning that. I'm certainly not questioning that. Some people will say that you should use a more aggressive regimen with doxorubicin and ifosfamide, and that does give you a better PFS, but the overall survival is probably very similar, and it's terrible quality of life while you're on the combination. So at most institutions in the United States, if you have widely metastatic sarcoma and there's no thought about resecting a oligometastatic disease, et cetera, you're going to get single agent doxorubicin. And you can see the PFS for single agent doxorubicin is not what we want. It's 6 months or less. And the response rate is not what we want. It's 20% or less. This is on all of the studies, not all of the studies, but some of the major studies testing doxorubicin versus doxorubicin plus another agent. So we did a study of doxorubicin plus pembrolizumab, the PD-1 inhibitor, and we saw better PFS than had been seen on these prior studies, 8.1 months. We saw better overall survival, 27.1 months, and we saw some patients with very durable responses. You could see this going out more than a year with either stable disease or responses. And I've talked to some of these patients now who are 5 years out, who still haven't progressed with either dedifferentiated liposarcoma or undifferentiated pleomorphic sarcoma. So I think this is a really good regimen for some patients. And you could see in, if we just look at the undifferentiated pleomorphic sarcoma patients, these CT scans illustrate the fact that not all the tumor shrinkage is just coming from doxorubicin. So you can see here, after, this is a UPS tumor. After doxorubicin, it shrank, but the shrinking continued after 6 months of doxorubicin. Same thing with this dedifferentiated liposarcoma. And you can see that in each case, there were some patients who progressed, but 2 out of the 3 undifferentiated pleomorphic sarcomas had very durable responses. Half the dedifferentiated liposarcoma patients had durable responses. And this has since been kind of reproduced in another study from North Carolina. So we have now we have an upcoming big multi-center randomized trial that's we're finalizing the protocol with ECOG Akron. It's been NCI approved. It says under review at the NCI. The NCI has approved it, CTED has approved it, but the final protocol is still being tweaked. But I expect it'll be out in the next couple months. And this is going to be a big randomized trial throughout the US. And there's discussions that maybe even potentially elsewhere, looking at 160 patients randomized to either doxorubicin and pembrolizumab or doxorubicin alone. And I'm excited about this because I think it has the potential to change the standard of care. So let's talk about cellular therapy. This is one of my passions. This is kind of how I got into the field of immunotherapy of sarcoma. I was very interested in targeting some of these tumors with that express high levels of cancer testis antigens. So again, you guys have had some amazing speakers in this group. You may have heard about cancer testis antigens before. Cancer testis antigens, or sometimes they're called oncofetal antigens. That's like a new terminology for it, but they have a high level of expression in the testis. So this is the seminal vesicle. These are the precursors to sperm. So you have mature sperm in the middle of the seminal vesicle, the immature sperm at the outset, and the immature sperm express high levels of cancer testis antigens. There's also fetal organs like fetal kidney that will express, and fetal ovary that will express high levels of some of these cancer testis antigens. But most, and they can be expressed by placenta as well, but healthy tissues other than the testis and placenta in kids or adults do not express these. So there are good targets for cancer therapies. Some of the famous ones are NY-ESO-1, that's CTAG1B here. And you can see that the synovial, we looked at a variety of different sarcomas using a panel that we made with a lot of different cancer testis antigens. These, you can see a lot of them express MAGE-A9, a lot of them express MAGE-A4, a lot of them express PRAME, a lot of them express NY-ESO-1. So we were very interested in targeting these. Synovial sarcoma had already been described to express NY-ESO-1 when we did this study. But we were really excited that when we took a deeper look at myxoid round cell liposarcoma, every case that we looked at had some expression of NY-ESO-1. Since then, I think there are very rare patients that won't have any NY-ESO-1 or MAGE family antigens, but it's very unusual that they don't have even a little bit. And in most cases, it's homogeneous. So both with synovial sarcoma, in this study, over 70% of the cases, both synovial sarcoma and myxoid round cell liposarcoma generally have homogeneous expression of NY-ESO-1 and myxoid round cell liposarcoma and other cancer testis antigens in both synovial sarcoma and myxoid round cell liposarcoma. So I think there's been a lot of talk, and people get very confused about CAR T cells and gene-engineered TCR T cells. So remember, a CAR T cell, which is what's FDA approved for leukemia, ALL, lymphoma, these can be used for. They can be effective for CLL. They're very effective, these CD19 specific CAR T cells. And there are, of course, there's also CD20 specific CAR T cells. There's the BCMA CAR T cells and multiple myeloma. There's a lot of CAR T cells out there now, which are either approved or have some evidence of efficacy and are heading towards approval. These have an scFv, so that's like the part of an antibody that recognizes its target, and it's attached to a co-stimulatory molecule like CD28 or 4-1BB, and it's attached to like a CD3 zeta chain, which is what actually stimulates the T cell. And that will, when that recognizes a cell surface protein, the T cell will kill it. A TCR recognizes an intracellular protein that gets expressed on the cell surface by MHC. So you can see, and because of that, you have to have a specific MHC molecule, or a specific, in other words, you have to have a specific HLA type. And in the US, the most common HLA type is HLA A0201, which is why if you're going to try and target a patient with synovial sarcoma using a TCR, we require those patients to have HLA A0201, but there's no reason why you can't develop a TCR for HLA A24, which is the most common HLA type in some parts of the world, or any of the other class 1 HLAs, or even class 2 HLAs, right? And we've made TCRs for a lot of those, and there's companies out there that have TCRs against different HLA types, but it's a big production to make these T cells. Is very expensive for companies doing clinical grade production. It can be hundreds of thousands of dollars just to make one T cell product. That's not counting all of the costs that go into just treating the patient and your clinical research coordinators and getting the samples and doing the analysis to make sure that the T cells stay around, right? It's a big production. So far this has only really been looked at well in patients with HLA A0201. When I got interested in this, what I've always kind of been interested in is what the T cell repertoire is in the patient, and how can we make that more robust. So what I was doing is I was actually culturing out NY-ESO-1 specific T cells from these patients. So we would take a patient's blood, and you could see we use a reagent here that marks the NY-ESO-1 specific T cells in HLA-A0201 patients. And you can see this patient, you basically can't detect them in an untreated sample, but we would culture out dendritic cells, and we would pulse those dendritic cells using an NY-ESO-1 peptide. And then we would get a really nice population of NY-ESO-1 specific T cells, and we would use those to stimulate the patient's blood, and we would get, and that's what we would get, and then we would sort out those antigen specific cells, and we get a very purified NY-ESO-1 specific T cell product, and we would use those to treat the patients. And so we did a pilot with just these cyclophosphamide, with just these T cells using cyclophosphamide conditioning, and all these patients had some evidence of clinical benefit. One patient had stable disease for a year. One patient had an amazing reduction in the size of a liver lesion. One patient had some lung lesions that got smaller, but all the patients ultimately progressed. So they all had some tumor shrinkage, but they all had ultimate progression, which is kind of what's been seen on, I mean, some patients have durable, deep responses with adoptive cellular therapy. That's what you want. But most patients don't. Most patients will, even if they respond, even if they have a nice response, they'll ultimately progress. And that's really kind of the crux of the issue. How can we get these patients who have an initial response, how can we get them to have that response continue and be very deep? So you can see this is that liver lesion that I mentioned that had a nice response. This is a patient who went for resection of their lung lesions after they had adoptive cellular therapy and progressed. And this is staining for NY-ESO-1. And what you can see is that the NY-ESO-1 is still there. So they didn't fail because they lost antigen. So that's kind of something that can happen. Like in melanoma, there's lots of cases, not typical. I think it's overestimated how big an impact this effect has. But sometimes a patient will have, they'll be treated with MART1 specific cells, and they'll lose MART1 expression in the T cells. That's called an antigen loss variant. Most of the time, that's not what's happening, right? Most of the time, we don't know why these patients progress. Also, one of the big determinants of success in cellular therapy is the persistence of the T cells. So you can see these patients had persisting T cells, and you can see this is by flow. They still had a nice, though not big, they didn't have a big population, but they still had detectable T cells in their blood. So how is it that they still have a tumor that's expressing a lot of antigen and NY-ESO-1 specific T cells in their blood, and it's not eliminating it? And this is what drives me crazy, because I think that we act sometimes like we know, like, oh, we just need to have more T cells in the blood. Oh, their antigens are disappearing. They're not. That's not what's going on. It's something more than that. It's deeper than that. We need to figure it out. One thing that probably is a problem is that the phenotype of these cells that are adoptively transferred changes in over time while they're in the patient. So what we found is that so we did a bunch of sequencing and analysis of these cells that I'm not going to show. They don't have an exhausted phenotype, like they don't express PD-1, they don't express LAG3 or TIM3, but they don't also have markers of activation. They don't seem to be producing a lot of cytokines, and you can see that when we treat them with IL-15, they if you just have IL-2 in there, they don't grow very much. But if you use IL-15, they have a massive expansion. They also start to produce a lot more cytokines. And when we used an organoid model, these IL-15 treated T cells just totally eliminated tumors in the organoid model. These are T cells that we took from patients who had progressed after adoptive cellular therapy. So we took patients' blood, either from our trial, or we also took them from patients on a trial at UCLA or at the NIH. And we took their blood and we co-cultured it with tumor cells in an organoid model. And without IL-15, with just IL-2, the tumor cells, which are green here, still grew, and the T cells didn't do much. But if we used IL-15, the tumor cells were eliminated. So the other big problem, one big problem that we see with these tumors is they have low HLA expression, low MHC, and low T cells in there. So what we thought that one drug that is very old drug that we know can sometimes increase MHC and can sometimes increase T cells in tumors is interferon gamma. That's a very old drug, was tested in cancer for many years, never really worked in cancer, although there are some important clinical places where it works. Chronic granulomatous disease is a terrible immunodeficiency that children can sometimes be born with, and it's a drug for that. There's a condition called osteopetrosis that interferon gamma can use. So we were able to buy it, and now it's very expensive. But at the time, it was actually owned by a different company and was very affordable to purchase for a clinical trial. So we did a pilot trial. This is a phase 0 study where we did a biopsy of some of these tumors. We treated the patient with interferon gamma, and then we did a biopsy after 4 weeks. So it's a phase 0 study. A phase 0 study means that the primary endpoint of the study was a biomarker. So the primary endpoint was MHC class 1 expression. That doesn't mean that you're not trying to treat the patients, just like in a phase 1 trial where the primary endpoint is safety. You still want efficacy, but the primary thing you're looking at is the safety, right? Here, phase 0 trial, the primary thing you're looking at is a biomarker. So it's not safety, it's not efficacy. You want safety, you want efficacy, but the primary thing that you're looking at is a biomarker. So we're looking at class 1 expression. We can see here, at least in this patient, their tumor expressed a lot of class 1 after the treatment. Of course, that was not typical. So this is the patients. You could see more than half of them had a big increase in their class 1 expression. Some of them, now these ones that don't look like they had a lot of class 1 expression, we still think this may be important and relevant because they went from basically nothing to having something. So if you go from 0 or basically undetectable to having 1%, I'm not sure that is nothing, right? So 1%, of course, is not the same as 100% or over 80%, but 1% is still more than nothing. And this is a nice immunofluorescence. This is just gene expression on the tumors at the time of biopsy, showing that they had a profile more consistent with antigen processing presentation in the post-treatment sample. We also looked at NY-ESO-1 specific T cell responses in the peripheral blood. And we did find in 2 of the patients, we were able to show that they had a development of an NY-ESO-1 T cell response. So you can see here, these are PBMC treated with the negative control, whereas patients, the pre-treatment is on the left and post-treatment is on the right. And the post-treatment after treatment with NY-ESO-1 has a big NY-ESO-1 response. But we also saw an increase in PD-L1, particularly in the myeloid cells. So some of the patients had an increase in their tumor, and this is a striking one, but what we saw very consistently was an increase in the myeloid cells. So we had a trial of pembrolizumab, PD-1 inhibitor, and interferon gamma, but we did not see responses there. And we have some theories why now. So we're analyzing the samples in the lab right now, and I think we do have some clear answers for why this might be happening, and hopefully we'll be able to let you know about that in the coming year. We also tried interferon gamma along with the cellular therapy. So we gave interferon gamma for 2 days before our NY-ESO-1 specific T cells. We gave cyclophosphamide and then the T cell infusion, and then 2 doses of interferon afterwards. You can see this patient had a nice response in their lung lesion, but they ultimately, this is after the cell therapy, they developed this forehead lesion. So they did have technical progression, even though they were actually feeling a lot better after the treatment. So their pain kind of went away. They had shortness of breath, but that went away. They were able to go back to work. But this tumor was removed, and you can see it. This is post-treatment. This is the lung tumor on the right, on the left under D, and 10 weeks after is the forehead tumor on the right. And you can see NY-ESO-1 is still expressed, and E is the class 1 MHC, and that was not expressed afterwards. So there's definitely still more to do to figure out how to get interferon gamma to help these patients on cellular therapy. And two, the second patient had a terrible, tragic myocarditis that happened, and this forced us to stop the study. Although I will say this patient had very advanced synovial So this was a patient who had a terrible prognosis. They were treatment refractory. They were in bad shape, regardless of what we did. But it was so tragic because I mean, it was such a wonderful person with such a wonderful family, and it made me think that we really need to come up with some better ways to develop cellular therapy for these tumors. And one thing, and you can see now that this is only going to become a bigger part of the conversation. So MAGE-4 TCR is probably going to be FDA approved in the US. There are a lot of patients who have durable either responses or disease stability, but most patients you can see either have a response that's short-lived or who progress. So it's an amazing therapy. I'm so happy it's here. It's going to totally revolutionize our treatment for synovial sarcoma, but there's a long way to go in terms of making it everything that it could be. So the thing that we're really excited right now is the most common solid tumor in pet dogs is sarcoma, and there's a lot of really good data that the sarcomas that the pet dogs get are very similar to the sarcomas that humans get. For example, they get osteosarcoma, and their osteosarcoma is very similar to human osteosarcoma. They get what's called hemangiosarcoma, which is very similar to angiosarcoma. They get what's called soft tissue sarcoma, which is very similar to what humans would have as undifferentiated pleomorphic sarcoma. And all of these sarcomas express a protein called B7H3, which is a very popular target for CAR T cell therapy. There's a number of trials around the country targeting B7H3 with CARs. There's a company that makes a monoclonal antibody that's been tested in the clinic. So we made a canine CAR T cell using canine 41BB and canine CD3 zeta, along with B7H3 SCFB. And it's in right now. We've treated several dogs at the University of Illinois, something that's we're very excited about. And this, I think, is going to really give us it's not a TCR, so it's a different thing, but it's going to give us a way that we can try and improve cellular therapy using a really, because when you treat mice, the way, if you're making gene engineered therapy in a way where you're being tumor specific, and you're using real human tumor, then you have to be using a xenograft, and the xenograft does not have a human immune system. If you use a fully murine system, then you're losing some of the human element. But a canine tumor has a complex immune system that's much more similar to human compared with the mouse, and has a naturally developed tumor, right, that evolved through all the same steps that a naturally developed human cancer goes through. So we think this is a great system to develop CAR T cells. And this is Tim Fan, who we're doing this trial with in CAR T cells. Jeff Bryan is a veterinarian from the University of Missouri, and we're testing out some intratumoral injection therapies, specifically using IL-12 producing treatment. So hopefully that will even further increase what we can do here.